My name is Glory Martin. I'm going to prepare sample for extraction. When we receive sample, we cut it into small pieces for extraction, and we weigh it 1 gram in order to get the content which is needed per meter square. And from each sample, we prepare triplicate in order to get the average of content. So we are continuing to cut our sample into small pieces in order to weigh it the gram which is needed. We prepare 3 samples from sample in order to get average content of the sample. So we prepare 3 samples from our sample. Now we are putting our vial in weighing balance in order to weigh our sample. And we are weighing 1 gram according to the work instruction of Miranet. We tear the weight of the vial in order to put our sample.
So unataka ikwe
1 gram.
1 gram.
After weighing our sample, we'll record the weight because the weight we'll use in calculating the content and concentration of the sample. Now we are recording the actual weight of the sample which we are weighed. This is our sample 1. So we are preparing 2. After we prepare our samples, now we are going to put solvent in order to extract. In our sample, we are adding 5 ml internal standard and 45 ml xylene in order to get total volume 50, because the volume of extraction should be 50 ml. So we are adding 5 ml internal standard and 45 ml xylene. We are adding 45 ml xylene. Now we are tightening our samples in order to bring to water bath for extraction. And because the water baths have high temperature for extraction, we need to tighten it properly to reduce evaporation. Now we are setting the temperature which is needed for extraction, and the temperature needed for extraction is 88 degrees centigrade, not less or not more. So we'll wait until our water boil to 88 degrees centigrade. Then we'll put our sample inside the water bath for 45 minutes. But every 15 minutes we need to shake in order to get homogeneous solution. Eight. We are using stopwatch to set our time, 15 minutes for extraction, because the extraction time is 45 minutes, 15 minutes second time, and 15 minutes third time. So we'll set 15 times 3 times. I said we are extracting for 45 minutes, and it's already finished 45 minutes. So now we are removing our sample and we allow it to cool at room temperature. And after cool at room temperature, we are filtering and we will process it with another stage of analysis. Our sample is already cool at room temperature, so now we are filtering, because we are not allowed to inject sample without filtering, because our machines are very sensitive. We are not allowed to inject sample without filtering, because there is other compound which are not good in our column. So we need to filter in order to remain the compound which is only necessary to our column. After filtering our sample, we are going to load our sample in high performance liquid chromatograph machine. So now, because it's already filtered, we are going to load it. After filtering our sample, we are going to load in high performance liquid chromatograph. And our instrument is one of the latest instrument, and it can detect a very small amount of insecticide, and it can detect 1 part per million or even 1 part per billion. It's like it can take, if we are million people, it can select the criminal from million people, or it can select the criminal from billion people. So it's very efficient instrument. First we'll put solvent for washing, then we'll put standard for reference, then we'll put sample to know the amount of content. When we want to load our sample, first we'll put solvent for washing, then we'll put reference standard, then sample. Because if you want to know the amount of our sample, we need first to have reference standard. As I said, we are going to load our sample in high performance liquid chromatograph, and our instrument is made of 5 parts. We have pump, we have autosampler, we have column, we have fluorescence detector and UV detector. The function of pump in this instrument is to pump the solvent to the column, and the function of autosampler is to take sample and inject itself, and the function of column is to separate the compound, and the function of fluorescence detector is to detect compound, and function of UV detector is also to detect compound. Our instrument is connected to computer software, whereby the output of the analysis is displayed, and the peak area showing us the presence of active ingredient in the required amount. Finally, we have the printer, which we are using to print our report and to be signed by head of unit and given to our client. We have 3 peaks, and the first peak is for solvent, which we use for extraction, and the second one is for active ingredient, and the last one is for internal standard. And we are using internal standard to reduce errors. If we have errors from one sample to another, internal standard helps us to reduce the errors.
I never wanted us to.
In the analytical unit, we also have the main instrument we are using to analyze our sample. In analytical also, we have the second main instrument we are using to analyze our sample, and it's gaseous chromatograph. And the first one, which I mentioned, is high performance liquid chromatograph. And in high performance liquid chromatograph, the mobile phase is liquid. But in this second instrument we are using, the mobile phase is gas. And the sample which we are injecting this machine is those sample which cannot decompose when it's burned. It's not vaporized. In gaseous chromatograph, we have autosampler, we have injector, we have nitrogen gas, hydrogen gas, and air. The second main instrument we are using to analyze our sample in analytical unit is gaseous chromatograph. And the gaseous chromatograph is the instrument which uses gas as a mobile phase. But in high performance liquid chromatograph, the mobile phase is liquid. In gaseous chromatograph, it's made up with autosampler, autoinjector, nitrogen gas, hydrogen gas, and air. Gaseous chromatograph also we are using computer. In our gaseous chromatograph, after we prepare our sample, we filter, we come here to load, we put our sample in the autosampler, and we set the sequence and we put vial numbers. So the autosampler take the sample solution into autoinjector according to vial number. Then the autoinjector inject the sample itself into the column, and the column get the sample, then the separation starts from the column. The screen of gaseous chromatograph, what we are seeing here is the column temperature. It's going up because we already switch on our instrument ready to analyze our sample, and the column temperature which we need to analyze our sample is 240 degrees centigrade. In gaseous chromatograph, we are injecting the compounds which can be burned at high temperature and it cannot decompose.
Yeah?
Niache hii ndio baba.
Yeah. As long as it.
Ukishaikatika. Ukishaikatika. Angalia sasa inavyofanya. Robot.
Imetoka kidogo.
Ndio. Kabisa lazima. In analytical unit, we also check the strength of the physical strength of the treated fabric. Because our net stays for 5 to 6 years, we need to know the strength of the fabric. And if the strength is not good, we return back the samples to factory to prepare the new sample which have the needed strength. And we are using martingale to check the strength of the yarn. We are setting the cycle. We are starting with 100 cycle to 500 cycle. And when it reaches to 500 cycle, we know that the sample are in the strength which can stay to 5 to 6 years. Here we have the small walls, so we can check how this wall expand during the cycle. Here we set 500 cycle in order to check if until 500 cycle the hole still like this, because if the hole expand, it means the strength is not correct. Yes, cycle. So 500,000 cycle. 500,000.
When you are ready. 500,000.
Okay, as we see, we have 200 cycle here. It's already run, but the hole still is small. So this fabric, the strength is okay. It can be used for 5 to 6 years.
I want you to
Point.
Nitoe. Nifanye hivi.
the name.
Nafanya hivi. Hivi. Kidogo tu. Ngoja kidogo. Unaongea sio kama unasema.
All right, my name is Franz Peter Mdoe. I'm the assistant head of Vector Control Unit here at the ATRC, Africa Technical Research Center. At ATRC, the Vector Control Unit, we have about 3 colonies of mosquito species. We have Anopheles gambi SCS, we have Aedes, we have also the Culex species. The colonies are established for for research purposes, to study, develop various findings as far as these vectors are concerned. The Anopheles gambi SS mosquito colony, which is established here, the species is one among the mosquito species which predominate sub-Saharan African countries, and Gambi SS is just one among the 8 different species of the Anopheles gambi complex, because the Anopheles gambi complex is just a group of 8 sublime species of mosquito. Others are like Anopheles arabiensis, Anopheles quadriannulatus, Anopheles melus, Anopheles melas, Anopheles amharicus, and Anopheles coluzi. So the Anopheles gambi SS is the principal vector of the most lethal malaria parasite, the Plasmodium falciparum. So it's like a coincidence. The most threatening vector of malaria, Anopheles gambi sensostrictum, Diptera culicida, transmit the most lethal parasite of malaria, the Plasmodium falciparum. Of course, Plasmodium falciparum is just one, but there are other parasites of malaria, or other species of Plasmodium, like Plasmodium vivax, Plasmodium ovale, Plasmodium malariae. So the 4 different species of Plasmodium, but the most lethal one for human beings, this is the Plasmodium falciparum, which of course predominates sub-Saharan Africa.
Yes.
So Anopheles gambiae SS is the most infectious vector of the most lethal malaria parasites, Plasmodium falciparum, that predominates sub-Saharan African countries. Now the colony of Anopheles gambiae SS is developed like just other colonies for the purpose of research studies, so as to control malaria, a disease that predominates sub-Saharan Africa, a disease which causes death cases for almost 90% of all malaria cases across the world are coming from sub-Saharan Africa. And the threatened groups are the children under the age of 5 and women, pregnant women. So that's a base for the reason as to why researches are being done so as to control this vector mosquito. In vector control unit, the mosquito colonies which are developed here, we start from getting the eggs of mosquito, some of which are obtained from the field and some of which we obtain from insectaries which are established from other areas like TPRI and some other places. Then the mosquito eggs are being reared in water, distilled water. Then the eggs do hatch into mosquito larvae. The larvae, which usually undergo 4 different stages: stage 1, first stage, second stage, third stage, and the fourth stage. Then the larvae do pupate, change into pupa, and then the pupa pupates into adult mosquito. Now, the rising or rearing of mosquito in the lab is more or less similar to the way mosquito rise in the field. So under normal circumstances, every human being is a mosquito keeper. Because if you throw away the plastic bottles in our gardens and some other places where water can settle, these are good breeding areas for mosquito. So we need to control mosquito in all stages. Principally, the alleviation of malaria by controlling mosquito, it is a secondary procedure, starting from the one of dealing with the treating individuals affected with the malaria. So mosquito can be controlled by 2 means, either at the larval stage, or mosquito can be controlled as adult mosquito. At the larval stage, mosquito are controlled by using either larvicides, or mosquito can be controlled by eradication of the ponds or breeding sites of mosquito. And furthermore, mosquito in the field can be controlled by using biological means. These are, for example, the use of fish, which usually feed into mosquito larvae. Now, the approach to control mosquito at the larvae stage is to try to keep human beings a bit far from mosquitoes. Instead of waiting adult mosquito, control for adult mosquito, the approach to control mosquito at the larvae stage is better and even cheaper. And therefore, this is just one approach, controlling the larvae. However, adult mosquito can be controlled by some other means, like indoor residual spraying, and by using nets, the treated nets, we call them long-lasting mosquito insecticide nets. They can be used to also keep mosquito far away from human beings. So the aim of vector control unit is to establish the colonies for the purpose of research in order to develop the sustainable control means of mosquito and the alleviation of malaria. The purpose of vector control unit is to conduct research based on these vectors. Now, we usually collaborate with other institutions as well. We share up information so as to make our work even better. For example, like you are saying, the change in weather and adaptation of mosquito, as well as taking care of environmental pollution, the researches are being conducted. For example, to avoid environmental pollution based on synthetic larvicides, the researches have been done to use the botanical larvicides. The difference, one major difference is the synthetic larvicides, some are not target specific. They may sometimes harm even the non-targeted species like fish, and therefore to disturb the ecosystem. But the plant-based larvicides are target specific. They infect, they kill only the intended species. So if the larvicide have been applied in a pond or whatsoever, the target is mosquito larvae, and are the mosquito larvae which are going to be affected. Because the botanical larvicides, most of them are biodegradable, and they don't have some effects like bioaccumulation, and therefore they tend to be better than industrial or synthetic larvicides. The benefits of mosquito, one is they do balance ecosystem. Of course, yes, they might be harmful to human beings, but they are good prey for some other organisms like I mentioned before. And another benefits, they are used for the purpose of study, especially in schools, because they have mosquitoes are just insects like other insects in the phylum or the arthropod. So they are used for the purpose of study. And also diseases they cause, they provide employment. They do provide employment because professionals like doctors are being employed because of the disease which are being transmitted by mosquito. Researchers like I myself are being employed to research about mosquito. So it's a benefit. Okay, so just to mention a few, those are benefits of mosquito. Mosquito do not cause, but rather transmit.
Oh, transmit, yes.
Do not cause.
Yes.
They may cause, they may transmit diseases like yellow fever. Okay.
Is there a particular one that transmits that?
If I don't remember well, the Aedes aegypti. Yes. And then elephantiasis. If I don't remember well, Culex. Yeah, just mention a few. Yeah. Fever.
Fever.
Yeah.
Just like malaria.
You know, fever is a symptom of many diseases, because malaria and fever can be mentioned as two different things, because malaria is a disease while fever is a condition or symptom. Malaria is caused by the plasmodium, but fever, the rise in temperature. So someone might have a rise in temperature due to some other reasons, not necessarily malaria. Yeah. The control of vector mosquito is done in different styles, starting from control of larvae and then a control of adult mosquito. There are different approaches of controlling larvae, mosquito larvae, and there are different approaches of controlling adult mosquito. And the different interventions that have been done, some synthetic by using insecticides, some are synthetic insecticides, and some are botanical insecticides. So coming to your question, some of these insecticides which have been banned is because they rendered problem to the environment. For example, DDT is one of the insecticides, dichlorodiphenyl trichloromethane. It can harm the non-targeted organisms. That's one. But more ways, it exhibits bioaccumulation. The bioaccumulation, I don't know if you understand. Bioaccumulation, that essence by which they sprayed DDT, all right, after it has been absorbed by cells of an organism, all right, and if that organism has been consumed by another organism, the DDT, which was primarily consumed by the first organism, will still be taken by the rest of organisms. So this, of course, produce profound effects to other organisms as well, apart from the targeted one. Okay. Ideally, to get rid of these problems of the effect of non-targeted organisms on the environmental pollution and bioaccumulation, etc., etc., some of the solutions which have been employed is to use botanical larvicides. What is the benefit of botanical larvicides? The botanical larvicides, first of all, they are target specific because they kill what has been intended. Why do I say so? The botanical larvicides, some act like inhibitors. Inhibitor, a substance which hinder an enzyme to perform its role, and they are specific to particular enzyme, because an enzyme has got an active site to bind the substrate. So if the plant-based larvicide acts like a substrate and the enzyme is specific to particular substrate, definitely this insecticide is going to be target specific and is going to affect that particular kind of organism which is intended. And also, botanical larvicides, they are biodegradable. That means if they are consumed, they will never exhibit bioaccumulation because they will be broken down into different products. So ideally, the use of botanical larvicides have got advantages by far from synthetic larvicides, so to speak, based on the evidence of which I have given. Yeah, that's very good. Okay. Replicates?
Replicates, yes.
Okay. The assays, how many replicates? 20?
25 rabbits each beaker. And then 3 duplicates per. Can you help me?
Don't forget.
Then next.
Just 1 minute, please. One. Come down. Okay, please. Vinaenda juu, zinakaa juu. Here. Okay.
So these are ours maybe 48. Mortality is recorded. Essentially concentration, but this time you are looking at is if, and that one is done in replicates, as you can see. The replicates aim is to minimize errors.
Okay.
Okay. So for example, you have one concentration here. You introduce maybe 20. Love it. Okay. And then you cannot use one beaker or one beaker. You have to use several beaker like this. So 7 of them bring results which are slightly different. But now the average will give you the reality. Welcome to the Insectry of Vector Control Unit here at ATRC. This is a place where larvae rearing, mosquito larvae rearing is done. The mosquito larvae in plates containing water and several mosquito larvae. So we have several plates for mosquito larvae of different colonies. For example, what you see now is a colony of Anopheles gambi SS larvae. And you have also a colony of Culex and a colony of Irex. So this colony here is the colony of Anopheles gambi SS. And the exercise going on, which is being done by Madam Sarah, I think she's going to introduce her name. Your name is?
My name is Sarah Ayo. I'm a lab technician.
Yes. So Sarah Ayo, our lab technician, what she's doing now is harvesting of pupa. Pupa after the 4th stage of mosquito larvae, the next stage is pupa, where pupa will be collected together and then ready for them to change into adult mosquito. It is a bit interesting, this species of organisms, larvae in water and adult living in terrestrial life. So this is a colony for minerals. So the colony of Gambi extends to larvae, and also here we find this side is a colony also of Anopheles gambi SS. But as you if you look at the larvae of Anopheles, Culex, and Aedes, they show some slight differences. If you turn around here and see, the larvae of Anopheles gambi SES, they just lie horizontally on the surface of water, and they occasionally move down, but they just lie horizontal on the surface of water, and occasionally they move down and up, right? But the mosquito larvae of this colony here, the mosquito colony of Aedes are a little bit different. They usually do not lie horizontal. They stay pointed, right? They stay pointed in a slanted angle over the surface of water, and they usually run away down, right? Just immediately as they see anything coming across. The major difference is on how they orient themselves in comparison to the larvae of Anopheles gambi SS. While those of gambi lie horizontal on the surface of water, these ones, they stay in a slanted angle from the surface of water. Also, another difference is the larvae of Anopheles gambi are a little bit smaller than the larvae of Aedes. Now, the size of larvae vary with the stage. For example, here we find plates of larvae first stage larvae. We call it a first instar. They are too small to be observed. If you move close here, you can see some tiny things moving around. They are all larvae, but too small because this is just first instar, right? But as you move to second instar, third instar, and the fourth instar, the size of larvae also increases. Size of larvae also increases. In most of our bioassays, we usually prefer to use stage the third instar because of two major reasons. The first reason, the third instar larvae, they stay in a little bit more time than the first, second, and fourth. So if you introduce larvicide, you get more time for the larvae to be affected with the larvicide, right? Different from other stages, which some take 2 days. The third instar may take more than that, and therefore, if we do bioassays by using third stage, it is easier to get more positive results than the other stages. This doesn't mean that other stages are not used. No, they are also used, but the most preferred one is the third stage larvae. After fourth instar larvae, then here comes the pupa, right? After pupa, then you get to have the adult mosquito. And the stages from egg now to adult mosquito may take roughly 2 weeks. The 2 days, first instar, 2 days, and the fourth also takes a relative short time if compared with the third instar, which it takes a bit longer time.
Like how many days?
Like 4 days, 5. What? Pardon?
What marks an instar? You call it an instar?
An instar.
Yeah.
The size of the
How do you know
instar.
it has moved from one to the other?
Yes, the size tells. For example, if you look at this, the tiny structures of the tiny stage of larvae signifies that this is the first stage instar. There is no a clear-cut demarcation which can tell you that this is the first or second or third. The most criteria which you use, the number of days, can tell us since this is 2 days, then you shift to second, 2 to 3 days, then the third one, 4 to 5 days, then the fourth one, 2 days. So-called morphogenesis, right? Morphogenesis, then the change, increase in size, right, and change in shape. Because growth is taking place, this is increase in size. Not only increase in size, but permanent, irreversible increase in size. And there is also change in shape. This is now we call development, right? Because the structures are changing. For example, The fourth instar larvae change into pupa. This is development. But the increase in size, this is growth. So both they form we call morphogenesis, change in shape and increase in size at the same time. Yeah. Cages, right? We have cages. Yes, we are trying to get rid of those who try to escape. There's no room to escape here. So as you can see, we have cages, all right? We have the first cage, second cage, third cage, and if you look around also, we do have some other cages. Inside the cages, there are adult mosquitoes. Just move close, you can see adult mosquito. Okay, adult mosquito. Okay, now the pupa, which as you saw the harvesting exercise that was done, then they put in a plate or bowl which is covered by a net, so that when they emerge into adult mosquito, they never escape. Right? They are trapped, and then by using aspirator, we bring them inside this big cage. Okay, so they are taken care inside the big cage. Inside here, as you see, there is a bowl which contain there is sugar.
Yes.
All right. The aim of putting sugar is to make sure that they get some energy. Now, why are they being given sugar? After pupating, the mosquito, of course, they undergo breeding, where the female mosquito, the gravid mosquito now, which of course they've got eggs. For the eggs to develop, they need, the female mosquito, of course, Anopheles mosquito or whatever kind of mosquito, needs blood. Okay, and outside there we have rabbits, which we use to feed the mosquito. Now, after the mosquito have taken blood from rabbits, then they are brought inside here. So we provide them with sugar, so at least they can get some energy before going to the exercise of laying the eggs. You have the black materials here, black cloth, which is covering, and then you have the black cloth like there. We are trying to create artificial darkness for the mosquito to lay eggs, because if you put them in just light like this, mosquito, they are so smart actually. They like laying eggs where the environment is a bit dark. That's why we put this dark material, so that they can easily lay some eggs. We usually do the same for the species for Anopheles, sometimes during feeding. You know, they prefer sucking blood in the midnight. So sometimes when feeding them, we need to again put a black piece of cloth, so at least they can suck blood comfortably. Right? But because if you put light just like this, the assumption is daytime, not yet time for feeding. So if you put a black cloth, then they assume that it is night and ready for sucking blood business. Yes. Behavior. All right. You see, some which are the larvae, third instar larvae, which are not completely pupa, they are still moving here and there. But usually, pupa never feed anything. All right. They are enclosed in something like a cyst. All right. So they can develop and mature into adult mosquito. So here we have the pupa. Just 1 minute. Especially the glass. Do you know how they locate where you are?
Hmm.
Do you know how they locate where you are?
No.
You are in a room, you just got in a room,
Hmm.
and a mosquito is far in another corner, and you locate yourself in another corner.
Oh.
Just imagine if you close yourself everywhere, you just leave a nose like this.
Hmm.
A mosquito will just come there and start giving out this sound, like that. But, again, what you see here, this apparatus is called an aspirator, which is used in picking the mosquito from the cage, harvesting mosquito from the cage for the purpose of study. All right. So our technician is going to show us the way to. You pass the aspirator inside the cage. All right. You do that one carefully for the mosquito not to escape, and then you blow the air back. All right. You pick a mosquito of your choice. All right. For example, the time when you have taken a cage during blood feeding of the mosquito, then the one which feed on blood, the female Anopheles mosquito, then you need to pick those which are full fed with blood. Okay. Therefore, you come across here, you observe the gloved mosquito which which are full fed with blood. Then you use aspirator, you pick them, you pick them, you pick them. All right. Then you transfer them to a card like this, ready for hatching. Instead of taking the whole cage, because it includes male and female mosquito. So we separate the two by using an aspirator. If you come closer here, you see the mosquito which have been harvested from the cage. These. All right. So they've been taken from there.
And there's a stopper.
Yeah, there's a stopper here for sure.
Yeah,
Yeah,
when
this-
we wanted to do the testing, we are using this aspirator for collecting female only.
Female.
Okay.
only.
Yeah,
The others will not come.
no,
No.
because we know this one is male and female.
Mm.
Yeah.
Mm. So we are returning them in the cage.
Okay.
Mm. You just blow air out. They just go back in the cage. And then you simply close our cage like this, carefully not to wrap around the mosquito. A mosquito net is designed relative to the size of the cage, and it is wrapped around, and then the structure like this appear. Okay. In Insectry, the mosquito are usually raised on a specified temperature and the humidity. The temperature is around 28, and the humidity is around 78 up to 80. Right, so we have this for recording those two stuffs, right? Temperature and humidity. And things mosquito need warmth, especially the species which we have. So here we have heater. As you can see, you feel hotness inside here. It's because of heater which we have. And also we do have what?
Humidifier.
The humidifier, right? Humidifier to create a conducive humidity around, all right? A conducive humidity around. If you talk about humidity, the amount of water vapor in the atmosphere. I'm holding this equipment here. This is called counter, all right? If you get to read inside here, the reading is 110, all right? This is a counter which is used to count either when you pick larvae or when you pick pupa. It is difficult to count 1, 2, 3 up to 1,000. You might lose direction somewhere. So we use this counter. After picking a larvae or pupa, you click once. Picking, you click. So at the end of the day, you just read from the counter. It gives you exactly the number of the shifted either pupa or larvae. Especially for the assays, larvae testing, all right, assays. Sometimes in replicates, all right, you need to put a specified number of larvae. Say, for example, 20 per replica according to WHO regulations. So in each replica or bowl which is used for experiment, like we saw the other time, you need to put 20 larvae. So you can't do 20 larvae for 7 replicates. You might lose a point somewhere. So what we do is you pick a larvae, you transfer it to a test bottle, and then you click the counter.
ningetaka ukue kwa hali hii, kwa hiyo wacha amalize.